Before we start, we have to start with the introduction. First of all, we begin with the basics. We need to understand this word genetics. Then we now go into this one, Mendelian. So the word genetics just has to do with hereditary and variation. So this word now is divided into two. So, like we know, heredity, just like we say inherit, heredity is just the study under biology that studies the characteristics and traits you inherit from your parents and that is passed down from you to another generation. So that's just everything about heredity. I'll try to just break everything down. So now variation now it just studies the differences among human beings or even animals and plants, all the living things, the differences among them, because that is what we ascertain for the fact that a parent will give birth to two twins and two of them will be very very identical, but still they will have their different differences. It is under this part of genetics that we study. Now this word now Mendelian is derived from the name of the man that started genetics. He's known as the father of modern genetics. So and that is Gregor Mendel. So if you don't call it Mendelian genetics, you can call it classical genetics. So that is another name for Mendelian genetics. So genetics just studies those characteristics and traits that you got from your parents and you will transmit to another generation and the differences within those characteristics. So that's just the study of genetics. So and that is mainly what we study. So another thing I'll stress on is why do we study genetics? So the reason why we study genetics is very very important because it helps to identify genetic mutations that predispose individuals to certain diseases. Let me just stop here because I just want to use this thing to explain to people why we are meant to study this course in the university. So genetics helps you to identify genetic mutations. So mutations which causes variation, which is a kind of variation that exists between individuals. Sometimes it can be malignant, sometimes it can be benign. When you study genetics, you can note these genetic mutations, be able to know the ones that are malignant and the ones that are benign, and it will help you to make decisions about your overall health, especially when you want to get married, about the kind of children you want to have. So that is another thing. Then technological development, for instance now the use of genetic engineering to produce stuff like insulin. It is just from the study of genetics because it is there that you learn about gene, DNA, chromosome, how you are going to take the DNA of a human being, then you transmit it into a bacteria, then you get it out of the bacteria. Then for instance, another human being, let me say someone that is diabetic, and it is due to the fact that the person's system cannot produce insulin, because you know that it's insulin that helps to maintain the blood sugar in the blood. So if the person, one person is normal, the other person is diabetic, so you can use the knowledge of genetics to get the DNA of the person that is normal, then you transmit it into a bacteria called Escherichia coli. But so when you transmit it to the bacteria will replicate, producing more of those DNAs. Then you extract those DNAs as insulin supplements. You give it to the person that has diabetes, and that is how you cure such kind of sicknesses. So these are one of the reasons why we study genetics. Now I'm moving down into the basic concepts of genetics. that without this concept we cannot understand genetics. And there are three main basic concepts. There are many other ones, but there are three main. So these are the three main concepts. And as I'm explaining these three main concepts of genetics, I'll be going into the other ones, the other concepts that you need to know, the other terminologies and terms that you need to know to understand this course very, very well. This particular course, if you see this textbook and you start reading it, you might think that it's very, very difficult. But if you understand the terms and the concepts that is used in explaining stuff in the textbook, it will be very, very simple. So that is why we are going to be starting here. So for me to explain this, I would like to go back to those levels of organization that we learned. I know that you guys learned it. That is from cell to tissue, tissue to organ, organ to system, then system to the body or organism. So when you now come, this, let me say, this is the body of the organism. So when you now come to your body, you narrow down to the system, to the organs in your body, to tissue and the cell. Then inside the cell, you have the main component of the cell, which is the nucleus. Then inside this nucleus is where you now found these three things. And now I'm going to explain them. After getting to the nucleus, the next thing you get is chromosome, DNA, and then gene. So let me explain it starting from the DNA. So the DNA is just a kind of a molecule or substance in our body that helps to give information or certain code sequences that our body needs to produce protein. And of all the molecules in the body, we have carbohydrates, lipids, and proteins. Of all three of them, the most important one, the biggest one that you need to survive in your body is protein. Why? Many structures in your body is made up of protein. Enzymes, hormones, your nails, your bones, many tissues in your body is made up of protein. So this DNA, for your body to carry out the functions, there has to be protein, and it's this DNA that is the center of that protein synthesis. It gives instruction, or it has the instruction already, because you get it through heredity. Like your parents give it to you. When you were born, you have the DNA of your parents. So and it's this DNA that carries the instruction necessary for the production of protein, and this protein is necessary for life. Without those proteins you cannot survive. Now this chromosome now is just DNA. When the DNA is condensed, we just call it chromosome. But this DNA is not just condensed. You know the structure of the DNA is a double helix. What I mean by helix is just like spiral, something that goes like this. But in this case, it's a double helix. That is, it has two branches, and it's these two branches that goes spiral. So that is why in some textbook you now see something like this. So this is just how the DNA looks. So when you now get this DNA, and it is very, very long, it's very, very, very long, that it passes the size of the cell like times 100. It's very, very long. So you need to wrap up this DNA and condense it for you to get the chromosome. And the thing that you use in wrapping it up is called histone proteins. So when you get histone proteins, it's just like, let me say, this is the histone protein. You now get the DNA. I don't know whether you are seeing. This is the histone protein. you now get the DNA and start wrapping it around it to make it more condensed. So you get the histone protein, put it like this, for example now, the bundle of histone protein. Then you now wrap the DNA around it. So after everything, you now have a condensed structure that looks like this. And this the chromosome. So this is the one that is inside the cell, in the cell nucleus. So and it is a condensed form of this DNA. Now a segment of this chromosome is now the gene. I don't know if you don't understand, you can ask me questions. Okay. Now I said there are three main components, and there are the three I've explained now. Let me just read the explanation they wrote here now. Now DNA. At the heart of genetics lies the remarkable molecule known as DNA, or deoxyribonucleic acid. So that is the full meaning of DNA. So this is the full meaning. So which serve as a genetic blueprint carrying the instructions necessary for the development, functioning, and reproduction of all living organisms. The reason why they say it is necessary for development, functioning, and reproduction is because it carries the instruction that you produce the protein, and it's the protein that forms development, that helps your body to grow, your bones to grow, for functioning of your body and also reproduction. So that is the definition this textbook used. Then they say structurally DNA forms a double helix configuration. I've explained it, resembling a twisted ladder with alternating sugar and phosphate molecules. So let's talk about the components of DNA. So this is the structure I gave to you guys now. So and DNA is made up of units. So just like we said, an element is made up of atoms, the body is made up of cells. Those things are the units. So for the DNA, the unit is nucleotide. These are the units that make up the DNA. So, and the nucleotide is now made up of three components. Number one is the phosphate group. Number two is nitrogenous bases. Then the third one here is sugar, pentose sugar. So the phosphate group is just PO4 minus. If you want to know it. Then the nitrogenous bases now, we still divide them into four. They are still they are divided into two actually. Then the two now has two each. So this nitrogenous bases now, if you don't understand anything, just call me back. I'll explain it very, very well. It's now divided into purine and pyrimidines. Then in purine you have two, adenine and guanine. Then in pyrimidines you now have cytosine and thymine. But if you are dealing with RNA, this thymine will be replaced with uracil. So in DNA you have thymine present, but in RNA you replace it with uracil. So just take note of it. Now, I've told you guys that it's nucleotide at the unit of DNA. Then the DNA, the nucleotide, each of the units is made up of phosphate group, nitrogenous base, and pentose sugar. Now, the pentose sugar for DNA is deoxyribose. The particular pentose sugar for DNA is deoxyribose. And what I mean by pentose sugar is that it has 5 carbons. That's just it. Then there are actually two types of pentose sugar, because this DNA and RNA, both of them are called nucleic acids. So the pentose sugar you can find in nucleic acid is deoxyribose and ribose. The only difference between them is this deoxy. That is, D means absence and oxy is oxygen. So in this one, at a particular point in the structure, there is an absence of oxygen in it. But in ribose, the oxygen will be in that one. That is just the only difference between the oxyribose and ribose. So there is no that if you want me to draw the structure, I will draw it, but I don't think that is necessary for now, because we still do it in chapter 6 of this textbook. So but just note the difference between them is the absence of oxygen at carbon number 2. I say that ribose is a pentose sugar, it has 5 carbons, but at carbon number 2 there is no oxygen for deoxyribose, but there is oxygen for ribose. So let's continue. So we are talking about the components of the DNA, and I listed that it's made up of the nucleotide, the phosphate group, nitrogenous base, and pentose sugar. So let me just show you how the structure is. If I'm using this linear ladder-like structure now, let's just use this now. These ones that are here, the rungs of the ladder, is the nitrogenous bases. One might be adenine, the other one will be thymine. Then for this one, cytosine goes with guanine. This is called the complementing of the bases, like complementing structure of the nitrogenous bases. Let me just give you a mnemonic to remember it. Just know that purine is adenine and guanine, cytosine and thymine. But when you talk about complementing, adenine always complements thymine in DNA. But if it is RNA, I told you that thymine is replaced with uracil. So adenine will be complementing with uracil. So this thing you can use, the mnemonic you can use is, so apple is for adenine and tree is for thymine. So apple on the tree, then car on the garage, something like that. So because some of these things in the exam, you might mix it up. You might see they might give you option. They put adenine, because here now you see that this two is for purine, but here this two is complementing. They will give you these two options, and they ask you which of them is a complementary basis. So you will now be confusing it. You now think that maybe that time you not remember that this one and this one is for purine, this one and this one is complementing basis. You now go and choose this one as complementary basis. So just know that the complementary basis, you can use this mnemonic if you choose apple on the tree, car on the garage. So that is that for now. Then this part, the backbone of the ladder, is now made up of the phosphate and sugar. That is why it's called phosphate sugar backbone. But these ones are the nitrogenous base rungs. So they can call it rungs or anything. Just know that they are the ones in the middle. They are the ones that are horizontal. These ones are the ones that are vertical. So they gave an illustrative example of the significance of DNA is found in sickle cell anemia. So since it's in this book, let me just explain that one. So in sickle cell anemia, that is where you see the significance of DNA. And what happens there is that a particular they say this inherited disorder results from a mutation in a specific gene that codes for the protein hemoglobin. The alteration in the DNA sequence produces hemoglobin molecules that are abnormal, making red blood cells distorted in a shape that can lead multiple health complications. So you know that the DNA is the molecule that gives the instruction. It is the thing that arranges the proteins, the amino acid, I mean, that will form a protein chain. So when the DNA is coding for a particular protein, for instance, hemoglobin is a protein and it's found in the red blood cell. It's the thing that attaches the oxygen, that enables the red blood cell to transport oxygen from one part to another. So if the DNA is coding for this particular protein, hemoglobin, it is going to make sure that each of these amino acid must align to form it. If this amino acid is A, this one is meant to be C, this one is meant to be D, this one is meant to be E. For it to code hemoglobin, all of them must be A, C, D, E. If this DNA makes a mistake and remove this C now and put something like F here, why it's coding for this protein, that is why the hemoglobin will have abnormal shape. It will now have a sickle shape instead of a disc shape. That is what causes sickle cell anemia. And let me tell you, the name of the amino acid that is changed is glutamic acid, because I think they asked us that kind of a question in our exam. Yeah, in by 101, they asked us the amino acid that replaces glutamic acid, which causes an abnormal shape of the hemoglobin, is that, and they gave us options: valine, methionine, leucine, and isoleucine. So the amino acid that is meant to be here, it is called the position 6, the sixth position in the beta chain. So in the sixth position of the beta chain of this sequence that DNA is coding, that sixth position, number 1, 2, 3, 4, 5, 6, the amino acid that is meant to be here is glutamine, or you can call it glutamic acid. So the extra copy is now on that 21st position. You have 1, 2, 3, 4, 5 to 46. Then on that 21st position, you have an extra copy here. Do you understand it? Let me just use this thing now to explain. You have, let me say, 19, 20, 21, counting, 22, 23, 24, 46. Then here in this 21st position, you now have an extra copy. Yes, an extra 21 chromosome there. So there will be two 21 chromosomes. Let's just say that each of them is called the name of their number. This one will be chromosome 19, this one will be chromosome 20, this one will be chromosome 21. So there are two chromosome 21 in that person's body. That's why it's called Down syndrome. So,
is there any other symptom that can get if there's an additional chromosome, maybe in chromosome 19 or 20?
yes.
It causes different
when you start reading, I think chromosome 19, when there is another extra copy, it's called Edwards syndrome. They have many names for the syndrome. There is one that is Patau syndrome. They have Edwards syndrome. But sharp, when you are going through the days, you will see.
Even this albinism, are you this what is it called, please? Albino.
Albinism.
Albinism.
Yeah.
Is it an extra chromosome that is causing it or lack of melanin?
Is lack of melanin. But the amino acid that is meant to produce that melanin is the thing that is lacking. It's not actually lack of melanin, but amino acid, just like this one now, the glutamic acid is replaced with valine. But that one is not a replacement. It's kind of, the name of that amino acid is tyrosine, that is meant to code for melanin. So when this amino acid is not in your body,
it doesn't have anything to do with chromosomes.
no. So let's go to Mendelian genes. Let's break it down. The works of Gregor Mendel. So no need of going through the history of this man, because you can read that up and another one. So, but his experiment, he used a particular plant called pea plant, garden pea plant. That green beans he used when cooking, that is the particular plant he used in his experiment. So if you don't call it pea plant, you can call it green beans. Then the scientific name of that plant is Pison sativum. So this is the plant he experimented on, and there are many reasons why he used this particular plant. Number one is the chromosome pair. It's very small. It has only 4 chromosome pairs. Total number of chromosomes is 8. Human beings has 23 pairs, but this particular plant has 4. Another thing, it has a very short lifespan. So some of these things is one of the reasons. But let me not just waste so much time here. You can read those ones off. But if you still want me to go deep, I'll go deep. But let me explain these particular ones. So first of all, let's talk about the laws of Mendelian genetics. Basically, there are 2 laws of Mendelian genetics. That is law of segregation and law of independent assortment. So these 2 laws are very, very simple actually. You don't need to cram them to know them in the exam hall. And they are also very, very close to mistaking. But I'll give you what you need to differentiate them very, very easily. So these laws of segregation. So before I explain the law, we have already talked about gene. So a gene now for a particular character, let's say someone's height, it has alternative forms, 2 alternative forms, or 2 different forms. Each gene has 2 different forms, and they are called allele. This is the allele. So for instance now, for tallness, you have the one for tallness, dominant tall, and the one for recessive tall. So this, the person that is very, very tall, this person that is short. So these are the 2 alleles of this particular gene. Now, what this particular law is trying to explain is during gamete formation. When your body cells want to form gametes, the testes or the ovary, when they want to produce the gametes. So during that gamete formation, these alleles of one particular gene. So if it is someone that is very, very tall, that is purely tall, homozygous tall, it will be T. So when that your cell, your testicular cell or ovary cell wants to produce your gametes, it has to break into 2 to produce. Normally your body cell is 2N, that is diploid. But after meiosis, it will have N and N. Normally, diploid into haploid, that is gametic formation. So when that is happening, these 2 alleles is going to segregate. One will go to the other cell, the other one will go to the other cell. That's just the law of segregation. During gamete formation, the alleles for each gene segregate. Segregate means they break from each other. Such that each gamete carries only one allele for each gene. So, normally in each cell there are 46 chromosomes. Then in each chromosome there are genes in a particular loci. We call it loci. The particular point a gene is found in a chromosome is loci, plural, or locus, singular. So there are many genes for each chromosomes. So the gene that you are dark or fair, the gene that you are tall or short, the gene that your hair is brown or dark or albino color, anything. So all those genes in that particular chromosomes, they has their alleles. The reason why the alleles are 2 is that one of them you got it from your father, the other one you got it from your mother. So that is why when you own gene want to segregate, each of those alleles, one will go to one cell, one will go to one cell, so that when there is now fertilization, that one from this cell and one from the person will now fuse together to form another one gene with two alleles from both parents. So that is why there is segregation during gamete formation. This thing is very very important, a key word. The alleles for each gene segregate from each other such that each gamete carries only one allele per gene. Now the law of independent assortment now is when we are considering, when we want to consider not just one gene at a time, considering more than, because in this particular law we consider only height, that is ct. If you are considering both height and skin color now, two of them at the same time, so you say that they segregate. We let me see, you have tall and tall, then you now have dark, dark. Maybe this one is dark, this one is fair, this one is allele for dark, this one is allele for fair, this one is allele for tall, this is allele for tall. So, and they want to segregate during gamete formation. Must it be that this one, when it's segregating, must always segregate with this one to go to one gamete? You know, if this one will segregate normal, this one will segregate normal. Or let me just use something like this. Now, tall, short, dark, fair. So if they are segregating, is it a must? Is it compulsory that these two dominant will go to one cell, while these two recessive will go to one cell? So this law says it's not a must. That is why it's called independent assortment of genes. When they are assorting, when they are separating, this tall one can go with this one, and it can also go with this one. So anyone it goes with is good. So let me just state the principle. You understand it from there. So the law of independent assortment of genes says that genes for different traits assort independent of each other during gamete formation. State the gamete formation. So when they want to assort now, these two different, let me use three now. Tall, short, dark, fair. Let's just use fat, slim. So we want to consider these three genes is present in one chromosome. So, and the person, there is gamete formation, and they need to assort because they are many. We are not talking about separation. Assort is that this one will go, this one will go, this one will go before each of them now separate. So that is just the meaning of assort. So when they want to assort now, after assortment now, this one now is not a must that if this one is separating, that this one will come, then this one will come, then this one will come. Like all the dominants will come in one cell. It's not a must. So you can have this in one cell. You can have this in one cell. You can have this. So the way they assort is independent of whether it's dominant or recessive. That's just the principle.
So this R is now, is it for one chromosome or the both chromosomes?
You see this?
Yes. This is tall.
Three of them.
Yes.
It's in one chromosome.
That is one.
I told you that this is chromosome, right?
Mm.
Then in chromosome, you now have segments in it. And each segment in it, there might be hundreds of segments depending on the number of characteristics that you have. All the characteristics that make you a human being is these things that we call gene. Each of them is a gene. This one might be this gene now. And each gene has two alleles. So this one might be this gene. This second one will be this gene. The third one will be this gene. So that is just
But when it comes together with the other chromosome, the other 23 chromosomes, I don't get, is it that when they come together, what will then happen? Because according to what you are saying, this add is in one chromosome, that is the 23 chromosomes, sorry. Then when it comes together with the other one making it 46, what will then happen? Like,
That is when you are joining the gametes.
yes.
It will just normally, like, let me see, in another chromosome now, this first gene is located down here. The only thing this particular chromosome is doing, it will turn so that the position of this particular one in this chromosome and the position in another chromosome and in all the chromosomes, if it is the first position, all of them, the gene for turners will be in the first position in all of them. Like all of them will align in such a way that they will have the same loci. Yeah.
There's another one confusing me like this. It's a close question, but I want to ask it. Since it's 46 that makes one like an offspring,
No, 46 in one cell.
in one cell,
You have all the cells, you have billions of cells, you have 46 each.
okay, but 23 comes together now
Yes.
to make one.
But okay, it's only sex cells that have 23.
Yes.
Then the body, oh, that is all the
The
somatic
body,
is 46,
yeah.
46.
The 23 here is not pale, just 23
Only
for
23.
for the gametes.
I was thinking it's for the both of them, like from the both parents, that is making it 46 now.
They join now.
To make one gamete, Abi. That gamete they're making, obviously it will lead to an offspring. That offspring, does it mean that that offspring will have 46 chromosomes?
In itself.
Yes.
Yes.
Then when it now grows up, it now turn to 23, Abi. I don't understand.
When you now grow up, you now reach the stage of puberty where you start producing gametes yourself. So when you are now producing gametes, that your cell that you have, that is 2N, you undergo meiosis. You know we have two types of cell division. As you are growing, it's division that causes you to grow. So as you are growing, the somatic cell will just be dividing to produce 2N. But meiosis is just for gametes, your sex cells, and those ones will now produce N. And it is only these sex cells that are considering. We are not considering the normal cells in your body. Only the sex cells. So when this 2N is about to divide, to form this gamete, it has to divide the number of chromosomes. One will come here. It has two sets. One will come this way, one will come this way. So they're not trying to explain why it is dividing. How does it divide? They now told you for one gene that has two alleles, the two alleles will divide. One of the alleles will come to this one of the alleles will come to this one. That is the law of segregation. Then if we are considering, normally you cannot have one gene in a particular cell. You have many genes in a particular cell. So if you are considering those many genes, there is where it is now telling you that all those genes are going to assort independently and divide, separate to each gamete. So it does not mean that that is why I decided to use this heterozygous allele. That is TT and maybe BB. So this one now is going to segregate. Then this one will segregate. But this one segregates now. This one might now come to here. This one might not come to here. This one now might also come to this place. Then this one will go to this place. So it's just probability, and that is why we study probability in genetics.
So does it mean that this second law is not only restricted to trihydrate? Like it's not only restricted to three.
You have gotten there now. We do inheritance, genetic Mendelian inheritance. We have monohybrid inheritance, dihybrid inheritance, and polyhybrid inheritance. So this first one is just when we are talking about monohybrid. That is one gene. But this second law is for polyhybrid, dihybrid, trihybrid, tetrahybrid, pentahybrid, hexahybrid, anyone. So that is just it. It is in polyhybrid that we consider the second law. So they might ask you a question is in the exam. They will say maybe they will say the hybrid inheritance uses which law of Mendelian genetics? They put one, law of dominance, law of segregation, law of independent assortment. Then they put another option, none of the above or all of the above. So that one, you already know that it's law of segregation. So that is it. So let's now do the hybrid, dihybrid, and trihybrids. But you guys understood the laws. When we want to do these experiments, we can use two methods. We can either use the crossing method or we use the Punnett square. So I'll show you guys two of them, anyone that you now prefer to use. An exam, they might choose, but Punnett square is mostly used for polyhybrid inheritance. So, but you can still use it in monohybrid. So, for instance, now they said,
When we were studying, they gave one concerning three methods,
okay, the fork
and I
method.
did not understand it at all.
Is it this one? Fork line method. I will still explain it. It's still used for polyhybrid, but this is the two main. Even in your exam, it's OBJ that you can write, so you only come across this two. You cannot see this one. A question that will require this one in your exam. So let's now start with monohybrid. So in monohybrid inheritance, we're already studying inheritance when we are considering just one gene. But normally, it is not feasible because normally in your body you have many genes that shows for many characteristics and traits in your body. So, but we are now considering, let's just say we are studying one. And before I move on, Mendel, using the Pisum sativum, he studied 7 traits. Let me list them now. The first one is stem length. That is whether it's tall or short. The second one is pod shape. This particular question came out for us during one time. They asked us how many distinct traits do Mendel experiment on in the pea plant. So they now give you 6, 8, 7, 5. So you now choose your option. The answer is 7. So pod shape now, we have inflated pod and constricted pod. Then seed shape, we have round seed and wrinkled seed. Seed color, we now have yellow and green. These first ones I'm writing is the dominant one. Second ones are the recessive ones for pea plants. Then we have flower position. If you don't understand any of these ones, just let me know. Let me explain it. Flower position, axial or terminal. Then flower color. Purple and white. Then pod color. Green and yellow. So you can notice now the seed color and pod color is different. The dominant seed color is yellow for that pea plant. But you all know you used to buy green beans, the color that you used to see mainly is green. So that is why the pod color, the dominant one is green, while the recessive one is yellow. Then the flower color, we have purple as dominant and white as recessive. Flower position, axial is, let's just say this is the flower. The axial is the flowers that come out here. While the terminal is the flowers that come out up in the terminal. That's just different. So the ones that are axial is more dominant than the ones that are terminal. Then seed shape, whether the seed is round or wrinkled. Then we now have pod shape, the shape of that pod, whether it's inflated or constricted. The constricted ones are the ones that look like this. We just have something like this. So why the round ones don't have that kind of constriction? Then stem length, whether it's tall or whether it's short. So these are the characteristics that Mendel studied. So if I were to do monohybrid inheritance for this pea plant now, let's choose one of the genes. Anyone, you might choose stem length now. That is, we have the alleles, either this or this. Hope you guys know what is generations. The first filial generation, second filial generation, the parent generation. So let's say for instance now, let me use this book. Let's say we want to cross pure tall and pure short. That is homozygous tall and homozygous short. Now, for the parents, this is what it will look like. This a parent that is 2n, and this the other parent that is 2n. So, but before there is fertilization, the crossing is just showing you the fertilization to produce an offspring. So before fertilization can occur, the parent has to produce gametes. So if this thing is theory, you must show that the gamete part, that is something like this, something like this. That is this one gamete, this one cell produce these two gametes. That is law of segregation now. These things will segregate into these two in the gamete. The same thing here. So the thing you cross is the gamete and not the parent gene. So when you now cross the gamete, one of them will now come, you now have CT for the offspring. This is now 2n, this n, this 2n. Take note of this, because they might ask you the gamete, the parent, and the offspring, which one is which. Then this one will come, CT. This one will come, CT. And this one will now come, CT. So this is the result of the first filial generation. This parent and this gamete. If you don't understand anything, ask me now.
I want the 2n. Why do you write the 2n? Is it about the first ones?
The 2n is about the cell.
Okay.
This is the parent cell that will now produce the gametes after meiosis to form n. That is, the alleles is going to segregate into both cells. Then these two cells, one of the cells from one of the parents, let me say this is the father, this is the mother. One of the cells here will come, one of the cells will fuse during fertilization to form another diploid cell. That is why this one is 2n. So this is the first filial crossing. Now let's do the second filial crossing. So, but the conclusion from this one now, you cross homozygous pure, but you get heterozygous. This is one of it. So the ratio is 1:0.
1-0.
Hmm?
1-0.
Because we are considering two things, either it is tall or short. But everything you got here is tall. So that is why.
Is it the genotypic and the phenotypic?
Phenotypic.
Does it have phenotypic?
Phenotypic. Each phenotypic and genotypic is the same here, Sha.
Okay.
So both phenotypic and genotypic, the other one is still 1:0. But if you want to write it in genotypic ratio, it will be 0:1:0. Because first of all, you have to write the dominant one, then you write the mixed one, then you write the recessive one. If you are doing genotypic. But if you are doing phenotypic, you write just the dominant and recessive. So the first one is always dominant, the second one is recessive. So that is just it. And why I say it's 0:1:0 is because it is just the mixed one that you produced. So the other thing the textbook gave us is crossing the heterozygous versus the heterozygous, and two of them will be dominant. So okay, let me show you how to use Punnett square here. First. So if I have to use Punnett square here, Punnett square is just very simple. Let me just make it beautiful. So this is the crossing sign here. The ones, one of the parents will be here, one of the parents will be here. So T and T, T and T. So these two we need together. T and T. Just know that dominant always comes first, no matter the one that is here and here. T and T. T and T. If you don't get it, ask questions. Do you understand the Punnett square?
How about the top method?
The fourth line method. The fourth line method, I will show you that one when we get to the hybrid. So, so let's do the heterozygous crossing. Stay with this Punnett square now. For me though, in the exam, I like using this one. It's more simple for me to use than this one. Because before I was finished drawing these things, it will waste my time. But this one I'll just draw it and go. Because it's OBJ, it's not theory. So you now get the phenotypic ratio is now 3:1. Because 1, 2, 3 are dominant, only 1 is recessive. Dominant, recessive. Then for genotypic ratio, dominant homozygous first, that is 1. Then dominant heterozygous 2, and then recessive homozygous 1. So that's just the ratio. If you don't understand how to get the ratio, you can ask me question now.
Yeah. I don't understand.
How to get it? Now phenotypic ratio, you know the difference between phenotype and phenotype. Phenotype is the physical appearance that you see. So if you have these traits now, 2 persons with this trait or 2 of these plants with this trait, the 2 plants will be tall, the same height. But the thing is that this one of its allele is actually short. But because this thing is dominant, it will not allow the shortness to express. So it is only the tallness that will express. So for this one, the 2 of them are expressing. That is, it's when you go deep into the genotype, that is when we talk about this. But when you are just talking about phenotype, it's just what you see. 2 of them are tall, but only this one is short. So you count the ones for phenotypic ratio. You count the ones that are tall. 1, 2, 3 are tall. This one is not tall. So 3 dominant is to 1 recessive, the one that is short. Then for genotypic ratio, that is when we now consider these ones now. The homozygous, this thing, dominant, heterozygous dominant, and homozygous recessive. That's just how we say it. Homozygous dominant, heterozygous dominant, and homozygous recessive. So we just pick it. The homozygous dominant will come first, which is just this one. This one is also dominant, but it's heterozygous. Because when the 2 alleles are the same thing, it's homozygous, but when they are different, it's heterozygous. So heterozygous dominant is now this one. Then the recessive one is almost homozygous. Because you can never have something like this. You can never have something like this, although in some situation. Because it does not mean that if you have a character for tall and short, that it is always tall that will be dominant. Sometimes short can be dominant. But in such a case, you must use capital. If you want to make short dominant, you must use capital S for short and small s for tall. That's just how you do it in genetics. You cannot say that if short is going to be dominant in this case, you can never say that. You cannot write it this way. You can never say it. Small letter can never come before capital letter. Instead, you use the name of that short S as dominant, which is short. Then the other S will be the opposite, the recessive one, which is tall. That's just it. So we start with dihybrid. And what do I mean by dihybrid? We are taking 2 characters. We are crossing 2 characters or traits at the same time, 2 genes at the same time. So let's say we are using this one and this one at the same time. We want to cross them at the same time. So tall and the shape, round and wrinkled. If you notice now, I use R for round because it's dominant. Then this small r is wrinkled. If wrinkled is going to be dominant, I'll use W. Then I'll make the small W round. That's just it. So let us use 2 pure tall and round, then short and wrinkled. That is something like this. Crossing with so when you want to cross it, first of all, gamete formation. This is just the parent cell. The second thing is gamete formation by using the cross method. So can someone tell me why this is like this? Why did I not like CT circle it, RR circle it? I explained this thing using this second law. Okay? Yeah. But can you tell me why? It's actually second law because second law, everything about the hybrid is second law. So the reason is we are dealing with two genes here, and each gene is going to segregate. So there are two cells. This gene will segregate, one will come here, one will come here. Then this other gene will segregate on its own. That is independent assortment. Then one will come here, one will come here. That is why we have it this way. So when you are now crossing like this for the first filial generation, you now have T, T, R, R. But you know, normally, if it is pure, all the offspring is going to be like this. So all the four offspring you get is going to be T, small t, capital R, small r. So it means that all of them are still tall and round, phenotypically. But genotypically, that you now see that it is heterozygous, tall and round. So if you want to write the ratio now, it is still 3:1. No, it's still 1:0 for this first one. Now let's do the second filial generation. Because they are normally, if I finish this thing, they are meant to be formed. So still the same thing 4 times. So all of them are tall, which is all of them are dominant. None of them is recessive. That is 1:0. The offspring can cross them. So 2 of these things now. So first of all, gamete formation, and this is where we are going to apply the law very well. That thing I told you that in the gamete formation, it's not a must that it will be like this. So from this particular thing now, we can get 4 possible gametes. We can get 4 possible gametes from this thing. Let me show you now. We can get T, R, we can get T, R, we can get T, R, and we can get T, R. So these are the 4 possible gametes. Then for this one too, these same 4 possible gametes. But when you have more than these two genes, let me see, you have, like the assignment they gave you guys is 6 genes. There is a hybrid A, B, B, D, D, E, F, F. This is what they gave you. So you're not going to find how many ways, how many kind of gametes you can find that is possible. This is just probability. It does not mean that this cell is going to divide into 4 gametes. It will always divide into 2 gametes. But in those 2 gametes, it can be this one and this one. It can be this one and this one. Any one. Okay? I hope you understand. So for this particular one now, 1, 2, 3, 4, 5, 6. That is the hexahybrid. This 8. If you want to get the number of gametes, we have a formula for it. 2 raised to the power n. Because it's very, very meaningful. For you to start, or we can use 4 line method here too. But if you just want to do it direct, maybe they give this kind of thing and ask you how many possible gametes can we get from this particular thing. You just use this formula where n is the number of this thing heterozygous that is here. If there is homozygous, don't count it as n. For instance, if there is C in between this thing and it's just like this, you are not going to count C as n because it's homozygous. N is just the number of heterozygous. So we count 1, 2, 3, 4, 5, 6. 2 raised to the power 6 is 64. So you get 64 possible gametes. That is what you can get from this particular parent. But in these 64 possible gametes, it is only 2 of them that will form gametes. These are the possible gametes you can get, but only 2 will be formed. So any of them, it can be A, B, D, E, F, G, anyhow. So that is the thing. But when you are doing crossing, you are going to consider all the possible gametes. So these are the possible gametes.
And why didn't you write some when you are listening?
So that is the exact question in the assignment.
Okay.
I was just using the assignment. So if you are to use fork line method for that particular, or for this particular one now, let me use the simpler one to start fork line method. Fork line method now, you start with one of them. Let's say T. T will branch into two. That is R and R. Then you do for the other one, T, which is still branch into two. R and R. Then you now have the four possible gametes, which is TR, 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 TR. It will look simple now because it's just two genes that we should do. But let me use that particular assignment now, using fork line method. But it's not important that fork line method must look like this. Oh. Some people used to use this thing. Anyone you want to use. So let's consider that particular thing. So we have A, B, B, C, C, D, D, E, E, F, G, G. So, and it is only one that we are going to make the master. But you know the genes now. It's your assignment, so you know that one.
I don't see.
You mistakenly wrote C. This one you wrote close.
Oh. So if you want to do this one, it's only one that you are going to make the head. Then the other ones will just be branches. So you are going to do for this capital A and do for small a only in fork line method. If you go to capital A, it will form two branches. Let me make it large at the beginning. The two branches is B and B. Then this B will form two branches again. D and D. D will form two branches. This one will also form two branches, D and D. This one will form, this one will form, this one will form. That is E and E, E and E, E and E, E and E. This one will form, this one will form. So it's somehow stressful, sha. Shall the assignment of genes now? For that is tetrahybrid. A, B and B, C and C, D and D, D and D, C and C, D and D, D and D. So this is where you have your outcomes. Each of them will have the outcome. So for the first one, you trace it like this. A, B, C, D. Second one, you trace it. You reach here, you go this side. A, B, C, D. Third one, you trace it. Since you have done this one, you are continuing here. A, B, you now go C, D. That is A, B, C, D. This one will now be A, B, C, D. Then from here, A small b, A, B, C, D, A, B, C, D. Then this one will now be small c, A, B, C, D, A, B, C, D. Then the second part of it, you just replace this capital in small a. So the other part goes, you just need for capital. It will now be A, B, C, D, 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 and A, B, C, D. So for this particular one, tetrahybrid, you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. That is 16 possible gametes. If you still use our formula now, that is AA, BB, CC, DD. You will still get 2 raised to the power 4 because there are 4 heterozygotes. They still give you 16. So when you're in the exam hall and they just ask you, they gave you this kind of thing, because I can still remember one of our questions. They just gave us something like this. That one is pentahybrid. A, A, B, B, C, C, D, D, E, E. Crossing with something like A, A, B, B, C, C, D, D, then E, E. So you cross it. Then after crossing the first generation, then the second generation offspring, you now get the possible gametes for that one. Or one of the questions, they say for parent A, they call this one parent A, they call this one parent B. They ask you, calculate the number of possible offspring that can be, number of possible gametes that can be gotten from parent A. Then calculate the number of possible offspring that these two parents can produce. So to get it is very, very simple. You get the number of gametes here, times it by the number of gametes of this one, you get your number of offspring. How to get the number of gametes? Using the formula 2 raised power n. This one is out because it's homozygous. 1, 2, just 2. So just 2 raised power 2 is 4. So 4. That is 4 gametes for parent A. There are four possible gametes for parent A. Then for parent B, this one is out, 1, 2. Still 4 gametes. So the number of offspring you can get from these two parents, number of possible offspring is 4 times 4, which is 16 offspring. And let me just show you. If you break down this thing down now using that principle, since these two is same, so the offspring must get that one. Then you have B, you have C, you have D, you have E. Then the second one, A, B, C, D, E. Then we now go. Am I forgetting anything? Okay, let's do this two now. B, D, E, B, D, E. So this A will go into this and into this, and like these ones are constant already because they are homozygous. This and this. So A, C, and E, you just go through like this, go through like this, go through like this, go through like this. So we've gotten A, B, and capital D for this one. A, small b, and small d. So the other ones we've not gotten is capital B and small d. So A, and then small b and capital D. So these are the 4 possible offspring of this one.